minutes. Friends, I am very happy that Professor Sir Tom Blundell has agreed to give this talk for the students of Yuvaraja's College and faculty and students of Mysore University. You know, there are few things in biology which has revolutionized humanity. One is the discovery of antibiotics. You know, the other is vaccines, discovery of vaccines. The third is, of course, discovery of insulin exactly 100 years ago. It has saved so many millions of lives. The impact overestimated. And, you know, insulin is the second protein that was crystallized. But it was crystallized in the 1930s when people did not know how to solve protein structures. Then, of course, Max Perutz and John Kendrew worked out how to determine protein structures, and they succeeded in the early 60s. And immediately, the structural studies on insulin crystals was undertaken by Dorothy Crowfoot Hodgkin, and Tom was one of her first associates. And they very successfully determined the structure of insulin. You know, ever since then, Tom has been using crystallography to elucidate molecules which are very important in terms of their biological function. This has naturally led him to ask, how can we predict protein structures without the laborious means of X-ray diffraction? And he has developed methods to construct protein structures using their amino acid sequence. And his program, Modeler, has become world famous, and it is used by tens of thousands of people on a daily basis. You know, he wrote a book on X-ray diffraction in 1976, Protein Crystallography, with Louis Johnson. And that textbook, even today, you see, after 1976, there have been so many developments in crystallography. That book should have been outdated, but still it is a classic book. You know, it presents the kind of history of X-ray diffraction, and it's a beautiful book. And that is, you know, very standard textbook. His contribution to, you know, this modeler program was made to help drug design. And he also has developed methods through which you can design drugs for known protein structures. You know, most of the drug targets are protein structures. So how to design a drug for a protein, for a specific protein, this has been his passion. You know, protein structures, the function, the folding, and what residue goes where, how to understand the protein folding problem, and how to manipulate protein folding, how we control the protein function, has been his passion throughout life. And he is such a wonderful speaker. It is a festival to listen to him. More than anything else, he has trained so many Indian postdocs. They now occupy major places all around the world. You know, that's why he has come to India so many times. He has so many friends. No matter which town he goes, he will find one of his associates there. So it's always a pleasure to welcome Tom to India. And Mysore also he has visited quite a few times. And it's my pleasure to invite Tom to give his talk. I am sure you will find that his talk is more or less interesting because of lack of slides. Thank you, Tom, you can start.
Well, I feel very embarrassed. I feel very embarrassed that my slides are not showing, and I thank you for your patience. So I'll try to speak to them, but giving a talk without pictures for me is a very challenging aspect. You may not know that my grandfather was a professional artist, and I was brought up doing painting and music. And the idea of giving a presentation without being able to have music is acceptable, but without slides is really challenging. So what I was going to speak about today, I just moved my slides over, is the centenary of insulin's discovery, and talking about the revolutionary developments that have occurred over the past 100 years, in which, of course, Indians have played many contributions as well. So I will begin my story just to give a summary. 100 years ago, when insulin was discovered in the pancreas, and I will just say how it became obvious, even 100 years ago, that insulin was stored as crystals. And I will then tell you how Dorothy Hodgkin realized that insulin would be a good target to work on. After she worked in Cambridge, she went back to Oxford and started working on insulin in 1934. So the insulin work has been going on for nearly 80 years. And it's certainly been one of the pioneering areas. So I'm going to talk a little bit about that and how the insulin structure appeared and how it correlated so well with what we could see in the pancreas, where it was stored in the islets of Langerhans, and also the resemblance of those islets to the crystals that Dorothy used. I'm going to go on from insulin and talk about one or two other targets, including HIV protease, which is, of course, an equally important drug target. And I came about that many years ago now, 30, 40 years ago, and realized that it related to some earlier work that I published in Nature about the evolution of aspartic proteinase enzymes. So I'll mention the work on that. And of course, I can't give a talk these days without talking about the SARS-CoV-2 proteome. And that's, of course, been taking up much of our time as we've been locked up by the challenges of the disease. So I talk about that. And then I want to talk about the real revolutions in the subject. One is cryo-EM, and I will talk about cryo-EM multi-protein systems. It's going to be difficult without proper slides because the revolution in cryo-EM, which has occurred over the last decade or so, has allowed us to see single copies of huge complexes. And the most recent work that I've done involves DNA repair. And I'm sorry, I'm not going to be able to show the slides of the complexity there. But all of that comes together in drug discovery. We can use our knowledge of structures of proteins, hormones, enzymes, and these complex multi-component systems that do things like DNA repair. We can use those as targets and use the structures to guide us towards new discoveries. And that's what I've been doing for many, many years. But for the last 20 years, I've had a company and we have 2 drugs on the market from that. I'll tell you a little bit about that. But it all came out of thinking about protein structure and how we would design new drug molecules. So I'm very pleased to be invited to Mysore and to visit Yuvaraja's college. I would certainly like to do that in person, and I'll do a better talk when I have the opportunity of being there. I'm sorry about this presentation, but it's really, apart from being 100 years of revolutionary developments, it's a personal history of 5 decades of India-UK collaboration on my part. So as I said, the first time I went to India is almost exactly 50 years ago. And I think I've been almost every year and sometimes several times a year since. And the people I've got on the slide here are people like Siv Ramaseshan. I've got Vijayan, M. Vijayan, who worked with Dorothy at the same time as I did on insulin and has remained a very close friend. Kalyani, his partner, and Srinivasan, and many others, including Nagasuma Chandra, for whom I gave a talk quite recently, and that one worked. I'm sorry this doesn't. So the insulin was discovered a century ago, and Banting and Best, I'd like to show a slide from it's 101 years ago now, of Banting and Best in Toronto, with the dog that helped them in their analysis of the pancreas, and how they discovered the islets of Langerhans, and later people realized that the insulin that was going to be so important is stored in these granules. And the granules are really microcrystals, and they exist in the pancreas. And so all we were doing really is exploiting a crystalline form that is the biological storage form. And those crystals, of course, have been used to treat diabetics. And the reason that crystals have been used is that they take some time to dissolve, and you then have your insulin being dissolved over a whole day, for example, and you don't have to keep pumping insulin in. You can allow the slow dissolution of the insulin crystals, just like the granules, to produce the insulin over a period of time. So that's the 100 years ago, but in the early 1930s to 1932 to 1934, the great man J.D. Bernal started working on protein crystals. And Dorothy Hodgkin went from Oxford to Cambridge, where I am now, where Bernal was in 1932, and started working with J.D. Bernal. Of course, he's well known for all his very left-wing political views, and their views that I read while I was at school and have been very much an influence on me. But in the period 32-34, particularly 32, they realized that they had pepsin crystals, pepsin, the protein enzyme, and they put it in an X-ray beam to see whether they could see anything. And of course, they couldn't see anything then. And then Bernal and Dorothy Hodgkin suddenly realized that probably they had to keep it in a water environment. So they kept their crystals in microtubes and sealed them so that there was water vapor. And then in 1932, they got the first diffraction pattern of an insulin, a pepsin crystal at that time in a capillary. And they wrote to Nature, and they said, now that a crystalline protein has been made to give X-ray photographs, it's clear that we have the means of checking them, and by examining the structure of all crystalline proteins, arriving at far more detailed conclusions about protein structure than previous physical or chemical methods have been able to give. So 1932, about 12 years after insulin had been discovered, they started on pepsin. But then Dorothy left Cambridge and went back to Oxford and wondered what she would work on. And she realized that these insulin crystals were being used for the treatment of diabetics, and she thought she would start work on insulin crystals. So in 1934, she got the first diffraction pattern and realized that that would give the structure. I joined the laboratory around 30 years later. A little bit earlier, I went in for just a term to look and see what they were doing. But in 1964, 30 years after Dorothy moved to Oxford, I went to join her laboratory. And I discovered that after 30 years, still insulin structure was not solved, although colleagues were working on it. There was Guy Dodson and Eleanor Dodson and Margaret Adams. And then Siv Ramasesan, great Indian scientist, went to join Dorothy Hodgkin in 1964. So I met Siv Ramasesan at that point, just as I was beginning to work on protein crystallography. And I joined Dorothy Hodgkin a little later. And then in 1968, Vijayan joined. He is, of course, now rather unwell, and has been for some time, but he has been the president of your academy. So he's made huge contributions. And I'm also very honored to be a member of the Indian National Academy for quite a few years. So that's the background. The Hodgkin group had Indian connections, and we solved the structure of the insulin hexamer. And the first thing that I realized about it was that it was beautiful, and it resembled in many ways a church window with symmetry for 12 apostles in the case of a church window, but 6 insulin molecules in the case of the insulin hexamer. And so I was completely transformed by this. I had also realized that the insulin structure we had, which was really 3 dimers coordinated by zinc atoms, was absolutely similar to the EM pictures of the storage granules. So that was very exciting. And I have some lovely pictures that I would have shown you of Dorothy Hodgkin and Vijayan and Guy Dodson, Eleanor Dodson, and myself in the 1970s when we solved, just after we solved the structure in 1969. But I, being in Oxford, was also heavily involved in other things. That's always been my weakness. So first I was a jazz musician. And so in the middle of the night or late in the evenings, I was playing with modern jazz groups at parties and having a great time with music. Later, I got to learn some Indian music, Carnatic music, playing the veena. But I also got heavily involved in politics. And I took over the planning of the Oxford City Council. Dorothy got very worried that I was doing politics in the late afternoon, music in the night, and insulin in the daytime. And I realized I had to think about things and what I was going to do. And so what I decided to do was to take a year off and, amongst other places, visit India. But what I did was I left India and went across the Trans-Siberian Railway. Took me 7 days to get from Moscow to Khabarovsk. And there I went from there to Japan. And then I went further round from Japan down to Taiwan, Hong Kong, and then moved. But before I moved, I got very heavily involved in watching and listening to Chinese music, which was amazing. And then I traveled on further, first to Calcutta, and then ended up in Bangalore, so not too far away from Mysore. But my first visit in 1972 was to Bangalore, and there I found Vijayan, and we had lots of discussions and planning collaborations. Of course, I already knew Sid Ramaseshan and his wife Kosonia, and also Vijayan and his wife Kalyani. And so these were all very exciting times in 72. And they built a collaboration that Dorothy had established between Oxford and Cambridge and India. And I've maintained that all the time that I've been. I've not counted the number of Indians that have been in my group, but certainly they probably number at least 50. And so that's been very exciting. The other thing that I was going to show you was that while I was in India in 1972, I decided to find out about Indian music, and I bought a veena. I'm sure you know all about Carnatic music and the veena. I was going to show you pictures of it, but you know, and I started learning the veena with an Indian friend who was in Oxford. I took a veena back from Bangalore on the plane and managed to get it back to Oxford at the time. So my other passion for India has been in Indian music. So when I returned back, I wondered what I would do. I thought I had to get out of Oxford, otherwise I would spend my time playing jazz music and I would spend my time doing politics. So I left Oxford, and Dorothy Hodgkin sorted out a job for me immediately in Sussex University. And there was no structural biology of the sort that I was doing. There was some X-ray crystallography in the chemistry department, but I was in biology. And what I did is I started and set up an old diffractometer and started doing structural biology in the School of Biological Sciences. And it was an amazing time. I went in 1973, and by 1976, in the 3 years, I had one paper in Nature each year. So I solved the structure of glucagon, which is the yin and yang of blood sugar control, along with insulin. I worked out with collaboration with chemists how insulin would probably bind to its receptor, although at that time we didn't know very much about the receptor. And I also speculated about evolution of insulin and proteins generally. And that was because John Maynard Smith, who was in the School of Biological Sciences, had written a lot about evolution at the molecular level. And I started to think about it and realized that I could think about insulin evolution in terms of Darwinian or selectively neutral mutations, a mixture of the two. And so in the 1975, I published the first major paper on insulin evolution from a structural point of view. And this really got me thinking about an area which I've ever since been heavily involved, and that is asking about the evolution at the molecular level and at the level of molecular assemblies. So that is where I was. And when I moved to Sussex University and was doing that, I realized that all the experience we had on protein crystallography was just not written anywhere. And so with Louise Johnson, who was a student at that time with David Phillips, who became Lord Phillips, and they solved the structure of lysozyme. Louise Johnson and I looked at all the work that she'd done on lysozyme and we'd done on insulin, and we wrote the first comprehensive book on protein crystallography. It's got around 600 pages, I think, and it was very good for me because it meant that I had to think about other people's work and the history of the protein crystallography. But that book has been cited thousands of times, and so a lot of people, as you were saying earlier, have used it. But when I moved to Sussex, it was really not a very large structural biology group, and suddenly I found that I was being chased in 1976 to become professor in London University at Birkbeck College. And this was the place where J.D. Bernal, that I mentioned before, had been moved to when he left Cambridge. And so for the previous 20 years or so, he'd been head of department, but had just died a year or so before, and they were looking for a successor. So I suddenly found at the age of 34, I had 100 people in a very left-wing interactive department with all J.D. Bernal's history behind it. And also in the center of London, where I could go and listen to both jazz and to opera, which is my other passion. So that was an amazing 20 years from 76 to 96. But what I did was to start asking the question, if structure could be useful to all the companies that were producing insulin, what about the role of structure for drugs, not protein hormones necessarily, but small molecules that were binding to enzymes? And I'd been working on an enzyme called pepsin and its relative renin, which is involved in blood pressure control. And so I started thinking about drug discovery. And so I have some nice pictures that you can't see of the enzyme pepsin, looking at the pockets where the substrate peptide went and all the hydrogen bond structure. And I think that was almost the first time that anybody had taken a protease structure in such detail and worked out all the interactions and started thinking about drug discovery. And that's because I could target renin and do something about lowering blood pressure. But I also realized in 1978 that renin and pepsin must have an ancestor. And I predicted an ancestor that was a dimer. It was published in 1978 in Nature again. So I have many papers in Nature. I was very lucky. And in 1978, I started looking for this protein that was the dimeric ancestor of pepsin and renin, because I thought if it existed, it was probably doing something which I could target in drug discovery. But it took me 6 years to find it, or for other people to find it, and for me to recognize that's what it was. We found, in fact, in the HIV genome, which codes for 10 genes, at that time it was thought probably more, we found this protease, excuse me, that we've been looking for. And what I did was to predict the structure of the protease, which was be the protease that process the HIV polyprotein. So to do with HIV. And I realized that if I could get that structure and I could predict it, which I did, and I could use then it to target to do something about AIDS and HIV. A lot of people said that I was speculating. I was told off by many people that I should stop speculating about these functions of things like the HIV polyprotein and its relationship to pepsin. So it took me a few years, around 5 years, to get the structure of the HIV protease. And I realized it was just as I'd predicted. And then we started, and other people in other places in the world started around the same time, to target the HIV protease and to get a new drug for the industry to cope with HIV/AIDS. So out of a distant relationship, I found myself working at the center of another disease, HIV, and I thought I'd better do something about rationalizing all of this. And of course, the HIV protease developed resistance, and I soon realized that understanding resistance mutations was going to be also important in new drug discovery. And so I divided my work into two areas. One area was going from gene to protein, target protein, and then the next from protein to drug. And I started defining the biological space. And so I was going to show you one example of DNA repair that I worked on, which illustrates this well. And DNA repair is where the double-strand DNA, in this case, is broken. And there's a system called non-homologous end joining that joins the ends. And what I've done over the last 15 years, or nearly 20 years actually, is to work on non-homologous end joining, looking at synapsis, which is how you maintain the double-strand break ends so you can join them, then end processing so you can remove some extra nucleotides and just put them together quickly, and then ligation when you put them together. So I've spent a lot of time doing structures of all of those components, and I was going to show you many of the pictures that I did first by X-ray. We published the structure of the huge DNA PK, that's protein kinase, which has over 4,000 amino acids, originally in Nature in 2010, so over 10 years ago. And then we got the X-ray structure at 4.3 angstrom in 2017. But at this time, I was realizing that I had to use the cryo-EM resolution revolution. So I, in parallel, started working on cryo-EM. And I have to say that in both my company, Aztecs, which I'll tell you about in a moment, and in my lab, almost all of our drug discovery, almost all of our structural biology underpinning it is being done now by cryo-EM. I'm hardly doing any X-ray work anymore, which is a great shame in many ways. But we have beautiful structures using cryo-EM of this DNA PK, which is a protein kinase. It's got 4,000 amino acids, and I could only get it at 4.3 angstrom with X-rays, but I got it at 2.8 angstrom around 4 years ago in cryo-EM. So I've been able to get the whole structure eventually at 2.8 angstrom, much higher than the X-ray resolution. And I was going to show you pictures of this amazing structure where we have assembly of 5 or 6 components in the DNA repair system. And we have cryo-EM pictures of them all, and at amazing resolution. And I was going to show you pictures of my colleagues, Amanda Chaplin, Steve Hardwick, and Tonya Kefalia, who've been working on this system and with whom I've been talking even this morning about these structures. So the super complex can be studied by cryo-EM because you don't have to have crystals. And so almost all the work that we're doing now is moved over to cryo-EM. And so what I've been doing is exploring the biological space and trying to understand the structures and then go to drug discovery. Now, I'm probably running well over time, but we've written, as was said in the introduction, a lot of software. I was told off by other crystallographers, including J.D. Bernal, not J.D. Bernal, Max Prutz told me off for spending a lot of time writing software rather than doing experiments. I did both, but I wrote the program modeler that was mentioned, and that has around 13,000 citations now, just for one paper in General Molecular Biology. And that's a modeling program which goes to satisfy spatial restraints. And we also had software to recognize distant homologs by predicting the three-dimensional structures onto a linear sequence and writing software called Fugue to identify those. So the software that we've been using, been developing, has been used all over the world. So 14,000 citations on the best, but much of the other software we have over 1,000 citations on as well. And we've also developed a lot of databases, not only for cancer mutations, but also for tuberculosis. And so in my company, I've been doing studies on cancer, but in my lab, I've been working on tuberculosis and other mycobacterial diseases. So I was going to show you pictures of databases for the structural proteome where we've used our software to predict around 80%, well, 88% in the case of Mycobacterium abscessus. And we've also worked on Mycobacterium leprae, and there it's another example of an Indian in my group, Sandeep Chaitanya, who's running the research in the Indian based American leprosy mission. And he moved to my lab, and we now have one of the most active centers in leprosy research. And we're trying to get drugs. And here it's a very different situation because people hide the fact that they've got leprosy because of the stigma. And you will know that in India, but in Africa, several states, there's a lot of leprosy. And many people don't know that it's also in places like Brazil, in South America. And so 200,000 people having these diseases each year and dying, many of them. So hugely important. And then the other thing that I've done is to move to the SARS-CoV-2 and take the genome sequence of that and build three-dimensional models for all the different components and start looking at not only drug discovery in that area, but also mutations. And we published about a year, almost a year and a half ago now, soon after the sequence of the SARS-CoV-2 became available, we have modeled all the components, and we published a paper on that and in briefings in bioinformatics. But more recently, the different components of SARS-CoV-2 we've defined using cryo-EM, including the NSP15 protein, which is one of the larger complexes with 3-2 symmetry. And we've got components using cryo-EM of the virus so that we can use them in drug discovery. at 2.4 angstrom in this case, but very high resolutions. Shireen Thomas, who's Indian from Kerala, and Shikan Liang, who's a Chinese, have the people been doing the most recent structures on this. And we've also written a lot of software. And of course, the great fashion at the moment is using AI. And so we've written a lot of software now for model building using machine learning in different ways and looking at ligand binding. So these are all things that many people are doing, but they all contribute towards understanding the targets of making new drugs. And we've been doing the same thing, as I said, not only in the virus, but in cancer. So in the academic lab, we've been doing some databases for that as well, for the 700 major genes of the cancer gene census. So that is the sort of background of the sequence and structure. We then, in around 22 years ago, developed a new method called fragment-based discovery for targeting these three-dimensional structures using tiny fragments of drugs, 300 molecular weight. I've talked about this in many places in India over the last 20 years. But with a library of around 1,000 fragments, you can screen a protein and then elaborate the fragments using structures to guide you. And so this is the basis of my company. A lot of people said it was totally speculative. We founded it in my lab and in the chemistry lab of Chris Abel. The key person was another Indian, Haran Jyoti, who'd moved to the UK when he was 8 years old. And he was in Glaxo Wellcome, and we had meetings on Sundays together, and he decided to leave, and we would start this method of using fragment-based methodology. So all I can say is that we got a company formed, a large building. By 2012, we had a pipeline. 2013, we sold the company, but they kept me involved. We sold it to a Japanese company for $886 million. But with it, we had drugs in clinical trials. And soon after that, we got our first drug for breast cancer, and then another one in 2019 for metastatic urethral carcinoma, and now continuing on many in cancer. So that's in the company, and I'm still heavily involved, although we sold it. But I then, in parallel to that, have been using these methods to target tuberculosis and leprosy and the infections of cystic fibrosis, one of which is Mycobacterium abscessus, which is, of course, very closely related to tuberculosis. And in parallel to that, we've been looking at difficult targets for cancer, because most of the companies have just been looking at kinases. And so in tuberculosis, what we've done is to attract the attention of Bill Gates. Actually, Bill Gates came and we talked to him about using our methodology, which he already knew about, to work on tuberculosis. And this was in the early 2000s. And Bill Gates then decided to put money into my laboratory to start up tuberculosis. He came with his wife, and Bill Gates was just an amazing person, as indeed was his wife. I'm sorry to find that they're split up now. But he was very interested in funding something for Africa, which the big companies pharmaceutical companies wouldn't find profitable. And so we started that in the lab. And of course, this was very good for me because tuberculosis was very, very important in Matabeleland in Zimbabwe, where my wife came from. She was born and moved when she was 19. And so we used to go back and discuss the designing of drugs for tuberculosis in South Africa and in Zimbabwe. And we used the same methods as we use for cancer, using different kinds of screening methods, thermal shift, SPR, and so on, and also getting X-ray structures of the different fragments and assembling those into drug-like compounds. And Bill Gates came again to Cambridge and visited our lab, and it was just amazing. He messaged me to say, Tom, don't tell anybody that I'm coming because they'll take me away to meet the vice chancellor of the university, but I want to see your lab. So we got him in, and he came on his own, and then we put him in a lab coat and had amazing discussions with him. And that's been very much the case with the collaborations we've had with Bill Gates and his team. And so we've developed all these methods for tuberculosis, for leprosy. And the other thing we've done, which I better not take any more time on, is to look at the emergence of drug resistance. So we've written a lot of software which can be used in cancer and HIV and in tuberculosis to look at mutations, as I indicated earlier on. And so we've been looking at the mutations that cause drug resistance in cancer. We've been looking at the mutations in AIDS and HIV protease and in Mycobacterium tuberculosis. And we've produced a lot of software. I originally, in 1995, with another group involving Srinivasan, who, of course, went back to Bangalore, and then another Indian, Arun Prasad, that I met and has now going to come and work with us after working in London for the last 10 years or so. And we've used statistical potential energy functions to predict the effects of mutations, but all software. And then the other thing we've done is to use machine learning AI methods to predict the impacts of mutations. These softwares are being very highly used all over the world. And I'm pleased to say that we wrote the software using machine learning between 2013 and 2019, before most people were using machine learning, as I think almost everybody is every day now, who has any interest in making predictions of any kinds. So that's the story. It's been a very exciting story where, as you can see, over the last 100 years, I've contributed to the last 60 of that, but inherited some of the enthusiasm from Dorothy Hodgkin. And for the last 50 years, I've been very much collaborating with Indians in many cities. And I'm very pleased that I'm a member of the Indian National Academy and get very well looked after when I visit. And we've been able to take part in these two revolutions, the two most important revolutions, I think, in the last 100 years, after those in the 1960s and 50s, are, of course, cryo-EM and machine learning. And that's where most of us, and I guess many of you, are working now. So that's where I am. I'm, of course, thinking not only about making new drugs to the unmutated proteins, but also repurposing old drugs and growing new drugs using our methods for when we have resistance. So that's my story. I have some pictures of many of my group, but I want to give credit to the fact that I think in the display of people I have, only 3 or 4, I think, are English. I have people from all over the world, and in the top line, I've got several Indians who've led various parts of my team over the years, but we also have people from Africa, South America, and so on. So my group is really multidisciplinary and multicultural, and that's been a wonderful way to run a group. And I think I owe guidance on that to Dorothy Hodgkin. And of course, Indian scientists have played a major role now. I can see quite a few Indians. I've mentioned Sheikh Arif, Pooja Gupta was another one, and lots of others in the group, Sandeep Chaitanya, and so on, who've contributed as Indians. So it's been a wonderful story, and thank you very much for inviting me to give this talk, and apologies for having no slides available for your access. Thank you very much.
Thank you, sir. That was very informative and insightful. Though we were not able to see it, we could actually imagine the story when you were describing it. And
I'm finding it very difficult to hear you.
Okay, thank you, sir. That was very, I hope I'm audible now. Are you able to hear me? Thank you, sir. That was very...
You are audible, but you are not clear. Speak a little slowly.
Sorry?
No, I said she is audible, but not clear.
Yes.
So I asked her to speak a little slow.
Yes.
Thank you, sir. That was actually very informative and insightful. And though there were no slides, we were actually able to imagine the story what you were trying to convey. So thank you for that. And I welcome everyone who has joined us today. And thank you for finding time visiting our webinar. My name is Sanjana Shajan, a guest faculty from Yuvaraja's College, and I'm your host for today's evening. And now the talk is actually open for questions. So if you have any questions, you can put it in the chat box. We will moderate it to you. Shreyas will be moderating the questions to the speaker. And over to you, Shreyas. Or if you want to talk to sir, you can raise your hand. So we will unmute you and you can ask the questions. So the talk is open for questions. So if you have any questions, you can put it in the chat, or you can raise your hand so that we can unmute you and you can directly ask.
Yeah. I think if it's a bit difficult with everybody spread around, you can always email me, of course, afterwards. Is there anyone there?
Madan is thanking you, sir. There's a question in the chat.
Yeah.
How much the serial crystallography can contribute to drug discovery?
I can't really hear you. Can somebody else just say it again?
The questioner is asking about serial crystallography. I don't know what it means, but how much serial crystallography can contribute to drug discovery? That is the question.
What kind of crystallography?
Serial crystallography. I don't know what it means.
I assume it means using shots over times, is it, to get the dynamics? I'm not sure what the questioner means, but that's the way we've used it, certainly. But I think we're very much better at structure than we are at dynamics. So serial is the best we can do.
Time resolved crystallography is the name of the crystallography.
There is a question, sir. How can we produce insulin in tablet form? By Dinesh Saroj.
Yeah.
Yes.
I'll try and answer any questions that come through by email, and that'll be fine.
Yeah, Devaki, I think you let them send email. You can proceed with the program.
Yes.
Okay. So we ask the participants to send the email for the questions, whatever you are having. So we will be typing the email ID of Tom, Professor Tom, so that you can directly send the questions to Professor Tom. Thank you. Sanjana, you can continue.
I really couldn't understand that at all.
Sir, the questions will be emailed to you. We will collect the questions. The participants will be sending the questions to you as you requested.
Yeah. If you could just send them to me, I'll try and respond. I'm sorry.
I think there is a little bit of audio glitches. So thank you, sir. And now I request Professor K.S. Rangappa, distinguished professor from University of Mysore, also former Vice Chancellor, to give concluding remarks. Over to you, sir.
Hello, everyone. It's indeed a special occasion for all of us as one of the eminent scientists of the world. And my good old friend, Sir Tom Blundell from the University of Cambridge is with us today. It's an honor to the University of Mysore to host him for a webinar. He delivered a scintillation talk on Centenary of insulin discovery. I must advocate that it is the right team to speak at this moment as we are celebrating 100 years for the discovery of insulin by Banting and Best.
Yes.
This is one of the remarkable discoveries of the previous century, as the whole world is dependent on exogenous insulin for the treatment of diabetes mellitus. Especially India is called as diabetic capital, and most of our population is dependent on either exogenous insulin or insulin sensitizers. Therefore, I can confidently make a statement that the discovery of insulin and other developments were maximally utilized by Indians. Although nowadays insulin can be produced either from swine or through genetic engineering approaches. Tom gave credits to original discoveries, that's Banting and Best, in his talk. So the effort of Dr. Devaki and her team in bringing such scientific giants in front of students is commendable and respectable. This creates a conducive academic atmosphere and encourages the students to get into basic research. And I'm sure the role of Professor Murthy in the growth of Department of Molecular Biology is invaluable. And I hope he will continue to support our institution in the same way in future also. I advise the Department of Molecular Biology to conduct more such scientific programs regularly. And I'm sure administrators of Yuvaraja's College and the University of Mysore always support such activities on the campus. I once again thank Sir Tom and everyone for allowing me to be a part of this program. Thank you one and all. Thank you, Tom.
Well, thank you. And I apologize again for having no slides available to you.
It was, yeah, it's descriptive and good. Thank you, everyone.
Thank you.
Now I request Professor N.S. Devaki to give us the vote of thanks. Over to you.
Thank you, Sanjana. Respected dignitaries and my dear students, it gives me immense pleasure to extend heartfelt thanks on behalf of the Department of Molecular Biology and Yuvaraja's College and myself during this occasion. First and foremost, I thank our special guest, Professor Sir Tom Blundell, Honorary Director of Research and Emeritus Sir William Dunn, Professor of Biochemistry, Department of Biochemistry, University of Cambridge, UK, for delivering a special talk on the topic Centenary of Insulin's Discovery, celebrating revolutionary developments in protein sequence, structure, and drug discovery. And I also express my heartfelt thanks to Professor M.R.N. Murthy, Distinguished Professor, Institute of Bioinformatics and Applied Biotechnology, Bengaluru, and AstraZeneca Chair and former Professor of Molecular Biophysics at Indian Institute of Science, Bengaluru, for introducing the speaker and for his presence. I extend my gratitude to our Honorable Professor K.S. Rangappa, Distinguished Professor, University of Mysore, for giving concluding remarks and for gracing this event. I extend my sincere gratitude to our Honorable Vice Chancellor, Professor G. Hemant Kumar, in his absence, for his continuous support for our program and our college. I extend my gratitude to our beloved principal, Professor Yashoda, Yuvaraja's College, Mysore, for her support in arranging this webinar. I also thank Professor Yuvaraja Gowda, Administrative Officer, and Professor K.B. Umesha, Control of Examination, Yuvaraja's College, for their support. I thank all scientists, faculties of different institutes, universities, and colleges for joining us. I also thank all research scholars and students for their presence. Our thanks are due to University of Mysore for providing the necessary facilities for the program and also for arranging live streaming of this program. Our thanks are also due to print media for giving coverage of today's program. I must thank the organizing team, volunteers, and all the participants for their active participation in making this webinar a successful one. I must thank the organizing team, volunteers of all participants for their active participation. I take this opportunity to remind ourselves of COVID-appropriate behavior and helping overcoming the pandemic at the earliest possible time. Please get vaccinated if you are not. Maintain social distance, wear a face mask, leave home only when necessary. Thank you one and all. Thank you, sir.
Thank you, sir.
Thank you.
Everyone here. And we have also put the email address of Sir Tom Blundell in the chat section, so you can copy it from there. We have also sent the feedback link, so please give us feedback, and we are sorry for the technical glitches we had initially. So thank you everyone, and we appreciate your time and for joining us. So until next time, we'll see you again in the next webinar. Thank you everyone for joining us.
Thank you. Thank you, sir. Thank you so much.
Thank you. Shall I leave? Yeah. Okay. Thank you.
Yeah.
Thank you so much, sir. So,
Bye-bye.
Cambridge,
Thank you all.
thank you.
I'm gonna kill